Hi students, myself Sudesh Kumar Kundidala, Assistant Professor, Department of Pharmaceutical Sciences, Vignan's Foundation for Science, Technology and Research, Guntur. In this video, we are going to discuss about the biosynthesis of pyrimidine nucleotides. So in previous video, we already discussed about the purine nucleotide biosynthesis. So that means we know that. So what is meant by nucleotide? So the nucleotide is nothing but the composition of nitrogen base, pentose sugar and phosphate. So in that nitrogen base, two types of nitrogen bases are there. So first one is a purine nitrogen bases and second one is a pyrimidine. In last class, we discussed about the how the purine bases are synthesized and what are the associated disorders and what is the regulation steps of this purine biosynthesis already discussed in previous video. In today's video or in this video, we are going to discuss about the biosynthesis of the pyrimidine nitrogen bases which are present in the nucleotides. Okay, so let us see that one. In coming to the pyrimidine biosynthesis, so we know that the pyrimidine structure, the basic structure of the pyrimidine is like this. So it is a heterocyclic nucleus. It consisting of two nitrogens in the ring system. Okay, so there is at third and first places there is a nitrogen. Nitrogen is present in the ring system, and the carbons and nitrogens which are present in the pyrimidine nucleus are obtained from the different molecules. So let us see how the pyrimidine nucleus is formed. So there are here also two types of biosynthesis are there for the pyrimidine synthesis. So first one is a de novo synthesis and second one is a salvage pathway. De novo means new synthesis, that means new pyrimidine molecule is synthesized. But salvage pathway means so it is utilized in the ribose molecule for the synthesis of it is utilizing the nucleoside. So there are nucleosides and nucleotides. Nucleotide is nothing but a combination of nitrogen base, pentose sugar and phosphate. Nucleoside means only nitrogen base and pentose sugar. So that means the nucleoside is nothing but without the phosphate. Nucleotide means the combination of nitrogen base, pentose sugar and phosphate. So in case of de novo synthesis, the nucleotides are built up from the pentose sugar. So that means on the pentose sugar molecule, the pyrimidine molecule is developed or it is constructed. So by obtaining the carbons and nitrogens from the different molecules. But in case of salvage pathway, by using the nucleoside, the nucleotide is produced. So there are nucleosides which are obtained from dietary source or it is available in our body. So by using that nucleoside, the salvage pathway will biosynthesize the pyrimidine nucleotides. Okay, understood. So in case of de novo pathway, it is a new molecule of pyrimidines produced by utilizing the pentose sugar. But in case of salvage pathway, by using the already existing nucleoside which is obtained from either dietary source or which is available in our body, so by using these nucleosides, it will produce the nucleotide. Okay, understood. Coming to the de novo biosynthesis of pyrimidine nucleotides. So these pyrimidine nucleotides are synthesized as same as that of the purine nucleotides, but the synthesis of pyrimidine is much simpler process when compared to the purine biosynthesis or purine nucleotide biosynthesis. Okay, and in case of pyrimidine ring or formation of the pyrimidine ring, generally aspartate, glutamine and carbon dioxide will contribute the carbons and nitrogens which are present in the pyrimidine ring. Okay, so but in case of purine, there are different types of molecules are required to contribute the carbons and nitrogens which are present in the purine nucleus. But in case of pyrimidine, only aspartate, glutamine and carbon dioxide is sufficient to contribute the atoms which are present in the pyrimidine ring. Okay, so this pyrimidine ring is first synthesized and then it is attached to the ribose 5-phosphate. But in case of purine biosynthesis, on the ribose 5-phosphate, the construction of purine nucleus takes place. So in case of purine biosynthesis, what will happen? On the ribose 5-phosphate molecule, the construction of purine ring will takes place. But in case of pyrimidine ring formation, first the pyrimidine ring is constructed or synthesized, then it is attached to the ribose 5-phosphate. So that is the difference between the purine and pyrimidine biosynthesis. In case of purine biosynthesis, the purine molecule synthesis happen on the ribose 5-phosphate molecule. But in case of pyrimidine biosynthesis, first the pyrimidine ring is synthesized, then it is attached to the ribose 5-phosphate. Okay, and this is the difference between these two. And coming to the sequence of steps which are involved in the de novo synthesis of pyrimidine nucleotide. So in that, first one is a formation of carbonyl phosphate. So first one is a formation of carbonyl phosphate. As I already said that there are 3 substances which are required to produce or to contribute the different types of atoms which are present in the pyrimidine ring. So what are the 3 substances that are required? First one is a aspartate, glutamine and carbon dioxide. So these 3 are sufficient to contribute the atoms which are present in this pyrimidine nucleus. And in that case, in the first step of this pyrimidine biosynthesis, there is a formation of carbonyl phosphate. Carbonyl phosphate is the first formation. Phosphate formation is the first step of de novo synthesis of pyrimidine. So for the formation of carbonyl phosphate, it is utilizing the ATP, glutamine and carbon dioxide. So when you see in this starting step of de novo synthesis of the pyrimidine, here we can see the starting molecules are nothing but carbon dioxide and glutamine. So by using the ATP, carbon dioxide and glutamine can able to form the carbonyl phosphate, and this reaction is catalyzed by the carbonyl phosphate synthetase 2. That is an enzyme. So this enzyme will catalyze the reaction to convert the carbon dioxide, glutamine into carbonyl phosphate by utilizing the ATP. Okay, and second one is a condensation reaction. So the formed carbonyl phosphate in the first step will undergoes condensation with the aspartate molecule. So the third substance which is required for the contribute all the carbons and nitrogens which are present in the pyrimidine is nothing but the aspartate, and that will involved in the second step of this biosynthesis. So the formed carbonyl phosphate in the first step will condense with the aspartate. To form the carbonyl aspartate. So this reaction is catalyzed by the aspartate transcarbamylase. That is one of the enzyme. So that can catalyze the reaction to convert the carbonyl phosphate to carbonyl aspartate. So here we can see this. So this is the carbonyl phosphate, and this carbonyl phosphate will condense with the aspartate. Thereby it can able to form the carbonyl aspartate, and this reaction is catalyzed by the aspartate transcarbamylase. Is the enzyme. So that will catalyze this reaction. And in the third step, there is a ring closure reaction takes place. So this molecule, so this is undergoes ring closure reaction, and this is occurred by the loss of water and So when you lost the when this carbonyl aspartate lost the water molecule, and it will convert it to form the, that is a ring closure reaction will takes place, and it will form the dihydroorotic acid. Dihydroorotic acid is formed, and this reaction is catalyzed by the dihydrovoratase. Dihydrovoratase is the enzyme that will catalyze the conversion of carbonyl aspartate to dihydroorotic acid. So during this reaction, the carbonyl aspartate will lose the one water molecule. Okay. And coming to the next, that is the fourth reaction, that is nothing but dehydrogenation. Dehydrogenation, so that is nothing but removal of hydrogen atoms from C5 and C6 carbons. So here we can observe the when you observe this structure, there is a dihydroorotic acid in this C5 and C6. So see, here it is a C5 and C6. From this, the hydrogen molecules, hydrogen atoms are removed from this C5 and C6 to form the voratate molecule or volatates. There is a product which is formed during this reaction. So dehydrogenation reaction takes place after the ring closure. So during this dehydrogenation, there is a removal of hydrogens from C5 and C6 carbon, and thereby it can able to form the voratate. And this reaction is catalyzed by the enzyme that is dihydrovoratate dehydrogenase. Dihydrovoratate dehydrogenase. So this is the enzyme which will catalyze the conversion of dihydrovoratate to voratate. Okay. During this reaction, there is a loss of hydrogens from C5 and C6 carbons, and this reaction takes place in the mitochondria. So the conversion or dehydrogenation reaction is a mitochondrial reaction. Inside the mitochondria, this reaction takes place. So as I said previously, there is a difference in the synthesis of purine and that of the pyrimidine. So that means in case of purine biosynthesis, the purine nucleus is built up or formed on the ribose 5-phosphate molecule. But in case of pyrimidine biosynthesis, first the pyrimidine nucleus is synthesized, then it is attached to the ribose molecule or ribose 5-phosphate. Okay. So here, so up to the fourth step, we can see the formation of the pyrimidine nucleus. So voratate is nothing but the one of the pyrimidine nucleus, and after the formation of this pyrimidine nucleus, it is attached to the ribose 5-phosphate. So let us see that one. So in the fifth step, there is a transfer of ribose phosphate to this voratate. So this transfer, so the ribose phosphate is transferred from the so this is transferred from phosphoribosyl pyrophosphate. So the ribosyl phosphate which is transferred to the voratate, so that is obtained from phosphoribosyl pyrophosphate. So which is then it will be converted into vorotidylate. So thereby it will form as the vorotidylate, and this reaction is catalyzed by the voratate pyrophosphotransferase. Phosphoribosyl transferase. So this reaction is catalyzed by the voratate phosphoribosyl transferase. Voratate phosphoribosyl transferase. And next, there is a decarboxylation reaction takes place. So the formed OMP, that is vorotidylate, is decarboxylated to form the UMP. So that is nothing but uridine monophosphate. So the OMP, vorotidylate, is converted into, that is nothing, vorotidylate means vorotide monophosphate, it is converted to uridine monophosphate, that is a UMP. So this is the first true pyrimidine ribonucleotide. So UMP is the, so that is a, in case of purine, inositol monophosphate is the primary nucleotide in case of purines, but in case of pyrimidines, uridine monophosphate is the first or initial pyrimidine ribonucleotide. Okay, so then after it is subjected to the phosphorylation. So when the uridine monophosphate is phosphorylated to form the uridine diphosphate and uridine triphosphate with the help of or with the utilization of the ATP. Okay, and this then the formed uridine triphosphate. So when it is utilizing the ATP and it is subjected for aminated, that is aminated, so it will form the cytidyl triphosphate by using the cytidyl triphosphate synthase. And then after it is subject for the reduction of ribonucleoside, that is a reduction of ribonucleoside diphosphate to their corresponding deoxy diphosphate. So that is a ribonucleosides will be subject for the reduction, then they will form the deoxy ribonucleoside diphosphates. Okay, so when the ribonucleoside diphosphate is subject for the reduction, then they can form the deoxy ribonucleotide diphosphates.